And I think it was KQ had questions about motility versus growth. All right.
Thanks.
And so I asked a woman that works on algae, and she said that they are motile. They don't have flagella. They don't have cilia. What they do is they excrete mucilage, and that allows them to grow in the direction that they want or need or signal. Yes, so Spirogyra is a filamentous alga that has a spiral chloroplast, and that's why they call it Spirogyra.
Did you ask her why it was a spiral?
No.
Okay.
Sorry, I had a beer in my hand, and I thought I was doing pretty good with fertility. But no, I didn't ask about the shape of the chloroplast. And my computer froze, and for some reason it's taking a really, really, really long time to come back up.
While we're waiting, do you guys just come together like a bunch of like scientists, bigwig people, and like shoot back beers and ask questions about science?
Most of the scientists that I know of drink a lot. A lot, and it's not even close. The last National Academy member that I drank with got so drunk that he couldn't sit on the stool and threw up in the bar before the night was over. I mean, he's like an 18-year-old kid. It's like, you idiot, really? But, you know, he has 30 people working for him and has more money than he knows what to do with. But he also is Scottish, so maybe that explains the drinking behavior. But normally Scots aren't like that. They can drink like fish, and you would never ever know that, you never ever know that they were doing anything. But yeah, it's kind of surprising how much beer gets consumed. The Maze Genetics meetings are famous for just, you know, one alcoholic haze. There were two 70-year-old people that were arrested in the pool naked having sex.
No! That sounds like the Saturday Night Live skit when they're in the hot tub. It's like Will Ferrell and the other girl, the really old one. Y'all remember that? Oh my god, that's so. You know what I'm talking about? That one's so funny. Yeah, the hippies and like the accents and talking about their lovers. It's so funny. I can't believe I'm like the only one who's seen it. It's so funny.
With Will, you said with Will Ferrell?
Yeah, it's like an old Saturday Night Live skit with like Will Ferrell and the Debbie Downer
They
character.
talk real slow and real.
Yeah, like that. It's so funny.
All right, so when I specifically asked her about the Engelmann experiment, she said it was a combination of motility and growth that led to the accumulation of bacteria in the blue and the red region of the spectrum. When we look at electron transport, there are a couple very interesting things. Oh, come on, don't tell me you froze again. It took, I was here early, and it froze.
Guess me, I can't. So you said the Engelmann experiment is a combination of growth and movement that leads to
Detection of the action spectra in the blue and the red region of the spectrum.
Cucumber, cucumber, cucumber. I know that trick.
And something that I need to tell you is that we've got the antenna complex, and then we have a reaction center. In the antenna complex, what we have is something called resonant energy transfer. So with the interesting thing about energy resonance transfer is that energy moves from pigment to pigment to pigment to pigment. Often when it moves, it loses a small amount of energy as heat. But electrons do not leave the pigment they're associated with. In contrast, our reaction center has a true redox reaction where it is going to lose an electron that is excited and will enter the Z scheme, the electron transport scheme. So this is a true redox reaction, where it loses electrons. And the typical scheme that we've got here is we've got carotenoids and xanthophylls, either of which will donate energy to chlorophyll B, which funnels it to chlorophyll A, which funnels it into our reaction center. So P680 is the name of the reaction center in photosystem 2.
I have a question.
Yeah.
So you said the energy is transferred as electrons.
So I think we're back here, right?
Yeah.
Okay.
So you said that it transfers energy via electrons.
So energy is transferred. Electrons do not move.
Okay.
Okay, so what it's going to do is if this pigment accepts a photon, it's going to boost the energy state of one of the electrons. This pigment will keep that electron and simply transfer that boosted energy state to another pigment.
Okay.
Okay.
That was a good question.
So that's like, each one of those pigments would be like a carotenoid, chlorophyll A, chlorophyll B, and then like the reaction center would be the P680.
So yes. So
Okay.
but you know what we could have is a xanthophyll, a
Yeah.
carotenoid, chlorophyll B, chlorophyll B, chlorophyll A, chlorophyll A
Okay.
in our reaction center. Melanie.
Is there a way to tell how much energy is lost between pigments, or is it a non-necessary?
There's not an accurate way to tell how much energy is lost that I'm aware of. You can detect the heat, but how do you differentiate the heat from the photosystems from the heat from all of the other biochemical reactions that are occurring? There's some work with electron spin states, and the chemistry is complicated. And I still don't think they have an accurate idea of how the energy is lost. I'll look at some papers to see if I can find anything interesting, but I think the answer is no. There's really no good evidence for that. Go ahead again.
Sorry. So the pigment molecules, energy is transferring between them. Energy is lost, but the electrons stay. Do the electrons determine?
Some of the energy is lost as heat.
Yeah.
Some of the energy is transferred.
Right.
Okay.
Okay, but the electrons stay. Does this have anything to like do? Well, so leaves, when you see them, you're reflecting. You're seeing the color that in the spectrum that it wasn't being able to be absorbed, and it's being reflected back to you, right?
Or is transmitted through.
Or it's transmitted through. So if these are all different pigment molecules, right?
Mm-hmm.
do the electrons determine what color passes through, or?
No. Its proximity to the next molecule in the reaction center, that is the primary determinant. And the reason for that is that if you look at the spectra, so let's pick out the ones that are important. Beta carotene is this brown color. Chlorophyll A is this red color. Chlorophyll B is this other brown color, this one here. And chlorophyll A should be here. Okay. So let's start. We have a blue light photon that's absorbed by chlorophyll A. It passes it on to a xanthophyll just because it happens to be adjacent. The xanthophyll spectra is not here. You'll measure that in 2 weeks. Then it passes it on to chlorophyll B, which is here, which then passes on to another chlorophyll B molecule, which is here at this energy wavelength that it's absorbing. So essentially what we need to do is we need to have an electron energy state that is lower in energy, but not a lot lower in energy to allow electron transfer. And you can see how much overlap there is in the blue part of the spectrum. And then once we hit the red part of the spectrum, it's going to go to chlorophyll B at 640 nanometers, then to chlorophyll A, and then to our reaction center.
Thank you.
Jacob, did you have a question?
I was going to ask about the mechanism for the transfer of energy. Does it just reflect the light?
Well, it's quantum chemistry. So essentially what's happening is that you have a packet of energy that's moving between two pigments. The electron doesn't transfer the energy, but in some mechanism, the energy gets transferred and it excites an electron in the energy receiving pigment to an excited state.
And when you say that, you mean like it changes the valence shell or whatever?
Yes.
Okay.
So it goes from the ground state to something that's no longer
Right.
the ground state. And as those of you that have taken quantum chemistry realize, is that once you hit an excited state, there are usually 3 or 4 excited states that are just a little bit higher that the electron is completely comfortable in, and the only important thing is really the spin on the electron. Is it an up spin or a down spin? Because there's a parity rule where you can't have an electron go from a down spin to an up spin or vice versa unless you have fluorescence. And we don't see a lot of fluorescence, so there's the spin rule as well. And I suspect that I just told you more chemistry than you wanted to know about this. But yeah, yes. Jesse, thank you.
Is the reaction center always a P680?
No.
Or it could be anything?
We have a P600 reaction center. We have 2 reaction centers, right? So PS2 is P680 and PS1 is P700, which gets us to exactly where I was going with the next point. How did we figure out that there were in fact 2 different reaction centers?
Through experiment.
Yes, through experiments. In fact, an extraordinarily primitive experiment. Anyone remember from the text? All right, one of our photosynthesis heroes, in fact, one of our plant phys heroes, came up with, we'll come back, we've already talked about this, and this, and this, and this. One of our photosynthesis heroes discovered the red drop. Okay. And what he observed is that if you gave plants a broad spectrum of light, they were perfectly capable of using all wavelengths up to 700 nanometers. Okay. However, then what he did is something completely ingenious. How many of you have read Ralph Waldo Emerson? Melanie, like him?
I like his prose, but I think that he tends to dribble on.
Blah, 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 yes, entirely. Gabe, do you agree?
Who are we talking about?
Ralph Waldo Emerson, one of the great American
Naturalist?
writers.
I read a little bit of Naturalist.
But oh, just so painful to read because he was that smart that he wrote really complicated prose that had really complex ideas. And he's just hard to read. His grandson, Robert Emerson, came up with the Emerson enhancement effect. If you turned on light at a wavelength of 700 nanometers, you got a little bit of photosynthesis. Okay. Turn the light off. If you turned it on 680 nanometers, you got a little bit of photosynthesis. However, if you had both lights on, what you had was a synergistic effect with 10 to 15 times more photosynthesis being recorded. If you're interested in measuring photosynthesis, any idea how you might do it with very primitive apparatus?
Black bottle, light bottle.
Black bottle and light bottle?
Yeah. You can drop it in and see how much they produce.
Oh, okay. So how much of what they produce?
Yeah.
No. How much what is produced?
Oh, oxygen.
Oxygen, yeah.
Oh, like when you take the spinach discs and you put them each in containers and then you cover up some with foil away from the light and some near the light, right? And then
Okay.
you see how many bubbles it makes?
Yes. So you're measuring oxygen, exactly.
Okay.
So this is what Emerson became quite famous for, is the Emerson enhancement effect. But he did this in the 40s. What he was truly famous for was telling a German Nobel Prize winner that he didn't do his experiments correctly and his data was wrong. How many of you have ever heard of Otto Warburg? How many of you have heard of the process of respiration? Warburg was the person that discovered and characterized respiration, and he did this with the Warburg apparatus in one of the very first oxygen electrodes, and he did this in the 20s. And the interesting thing about it is that how many of you guys remember the think about it question that I gave you on Friday?
Yeah, about the oxygen and photons. How many photons does it take to produce a molecule of oxygen?
All right, how many photons does it take to produce a molecule of oxygen? That was the question I gave you.
Yeah, there's like 2 answers for that.
And there are, in fact, 2 answers for that. There's the correct one, and then there's the one that the German gave. And both are very interesting and both are very acceptable for the question. And so I forgot to actually post this, but I'll post it later today, but I'm going to tell you the answer now, so you might not want to go yet.
Yeah.
Okay. So this ended up being a bone of contention between Emerson and Warburg. Emerson at the time was a graduate student at the University of Illinois. Even to this day, the University of Illinois is a leader in photosynthesis research. But he was the person that essentially set up the University of Illinois in that position. And so this is a man that was very, very clever, very careful, very meticulous, and cautious as well. I mean, he could very well have been an accountant, but yet he became a plant physiologist. This man was so careful that he absolutely refuse to fly in an airplane?
So it's my
Well,
stepdad.
well, back then, no,
Because
back
it-
then.
It was a little more dangerous.
My stepdad will
Um,
take a boat everywhere he goes.
Yeah. So at any rate, they tried to figure out how many photons, both Warburg and Emerson, thank you, Emerson, tried to figure out how many photons there were required for the evolution of a single molecule of water. So, how many people have a guess?
Well, I looked it up.
Chris? 8. Okay, how many people came up with the same number, 8?
Well, I came up with 2 answers.
How many came up with a different number? What did you come up with?
Well, I didn't figure it out on my own. I looked and it said that researchers found that it was actually 12.
Okay. So, you gave me one of the 2 answers that I would have accepted. The theoretical answer, the correct theoretical answer is in fact 8.
And why is that?
All right. Let's see if we have. And the reason for this is that you need to, in order to evolve 1 molecule of oxygen, you need to split 2 molecules of water. In order to split 2 molecules of water, you need to have 4 electrons added. Okay? And that will split a molecule, 2 molecules of water into 4 hydrogens and a single oxygen. All right? Why is 4 not the correct answer?
So system 1.
What's that?
So system 1 also need 4.
Exactly. Because in order to carry out linear electron transport and to make it so that we've got electron flow, we also need 4 photons here. So 4 photons to split water into 2 water molecules into oxygen, but to carry out linear electron transport, we need 4 more. So the theoretical quantum yield for a molecule of oxygen is 8. Okay? The actual quantum yield, Christine, is
12.
10 to 12, depending on the physiological state. All right, and what's the reason for that?
Energy loss.
Excuse me.
Energy.
Surprisingly, electron transport is extraordinarily efficient, and there is not really much energy loss at all. I mean, it's one of the very few times where you can say lost energy is not a good answer for this question.
I'm trying to remember, is it accessory pigments?
All right. In order to balance the amount of NADPH with the amount of ATP that is needed, we have both linear electron transport and cyclic electron transport. So I'm introducing an idea that we're going to get to probably on Wednesday. And this allows the correct amount of ATP and NADPH to be formed to carry out carbon fixation in the correct ratios. Okay. So the correct answer is 10 to 12, and the reason for that is that the chloroplasts need to make a little bit more ATP, which they do by cyclic electron transport that's not shown here. So, Warburg, on the other hand, said that the correct answer was 5. Remember, this was the 1930s. And they, Warburg and Emerson got into this back and forth with copious letters. Emerson finally said, I'll tell you what, I'll bring some of my equipment and I'm coming to Germany and we're gonna figure this out.
Ooh, he's having to bring the work to you.
Because I don't know how you get 5. So he gets to Germany and Warburg shows him his apparatus and sure enough, 5 is the answer, spot on. And he can't believe it because he's done this experiment 100 times. So he takes his equipment that he brought. Kabam, the answer's still 5. Warburg was elated. The whole lab went out to the bar, problem solved. They come back and let me finish the story. We're not there yet. They come back after the weekend. And Emerson said, let me do this again. I don't believe the results. He does everything again. There's only one difference. He turned off the green safe light. And kabam. He was getting 10, 11 photons per oxygen molecule. Did the same thing with Warburg's apparatus. Again, 11 to 12. Spot on the money, which is what he had seen the entire time. And the problem is that green safe lights appeared green to your eye, but they were incandescent bulbs that had a coating which was green and allowed other wavelengths to escape. So you saw green, you thought it was green, you thought there would be no photosynthesis, but there was. And it gets us back to the very simplest thing that we teach you as scientists, which is do the controls. Measure photosynthesis with no photons of light, just the green safe light. Nobel Prize winning scientists didn't even think to do it. Melanie, did I answer your question?
Yeah, I thought about it.
You thought about it and came up with that answer?
Well, I was also thinking it was between like improper light usage and atmospheric pressure in Germany, because they might be at a higher altitude, which it affects quite a few things, like baking and
Breathing.
Yeah, both are at between 500 and 1,000 feet. Champaign-Urbana, Illinois, and Munich, Germany. Not that big a difference in altitude.
Champaign, Illinois. It's a cool
Champaign-Urbana,
place. Champaign,
Illinois. Talk about a backwater, but it is it's the mecca
Well, they're doing
for
science there.
photosynthesis research. Even today, it's still the place to go. Jade.
Did I hear you wrong when you said that Emerson said that all of this stuff was wrong?
Emerson said that.
You said someone said that all of their research was wrong. Or
Yes, Emerson said that Warburg was incorrect.
Oh, I didn't mean that his work was wrong.
Oh,
I'm like, why would you not know that? Okay.
He sat there and fought with this guy literally for almost a decade before going to Germany. And he went to Germany with all this stuff, and he took a boat. So let's get back to here. We've covered a great deal of information. Tell me why we observe the Emerson enhancement effect when we get both red and far red light. Blair.
Yes, because both both reaction centers are working.
Exactly. You are stimulating both reaction centers using these wavelengths of light. Why do we get any photosynthesis here? So this is only going to stimulate PS1. Why do we see any oxygen evolution at all?
Because of the cyclic transfer we were talking
So
The linear transfer.
about?
what's going to happen is that we're gonna use up all of the electrons in PS1, and we're gonna see a little bit of O2 evolution, and the same is occurring over here. So essentially, we're emptying the photosystems of all of their electrons, but we're not replenishing them.
So does that mean if you were to leave the light on, would it eventually photosynthesis cease once all the electrons were transferred out of the photosystem?
So if we actually had, if Emerson actually had monochromatic light that was totally clean
Mhm.
at the wavelengths I said, the answer would be yes.
Okay.
He didn't have access to light that was monochromatic in that sense, and there was a little bit of bleed over into the other photosystem.
Okay.
All right. So it has to do more with technological limitations rather than anything else. Because theoretically, you're exciting photosystem 1 and removing all the electrons from photosystem 1. There's not going to be any electron movement from photosystem 2, where we have all of our oxygen evolution coming from. So if he had done this, with monochromatic light, this would have been 0. And this would have been right where it is until you've depleted all of the electrons from all of the PS2 photosystems. And this would have been up here.
Okay.
And you still would have been able to justify 2 photosystems. But this is the rationale for the Emerson enhancement effect. And he used this to propose 2 photosystems, hence an expected quantum yield, which was exactly what Chris gave us, of 8. He observed 10 to 12, and his explanation was there was loss of energy. And perfectly reasonable explanation, but in this case, it wasn't correct. So it's the one thing Emerson was not correct with in his entire life. Jade.
So for the red light, it's photosystem 1, and then
Red light, no. Red light is PS2.
Yeah, but the far red.
Far red, PS1,
Yeah,
yes.
and then when you have both of them on, they combine and.
To get linear electron transport and a much higher rate of photosynthesis.
So by itself, it's cyclical, not linear?
So
Each one by itself, is it cyclical?
PS2
Can
can be cyclical.
be. PS1 can.
PS1 is not.
So does all PS1 go to PS2? Like are they consecutive?
Nope. In fact, it's a misnomer because we start